Hi, I'm Lestari and I'm gonna explain about genetic material. So let's get started. Let's start with chromosome. Each chromosome contains a long strand of DNA that carries genetic information and controls the activities. DNA forms a double helix. Within the DNA are special sections called genes for making proteins. DNA is made up from two strands. It's made up of lots of molecules called nucleotides. Each nucleotide contains three different parts: a phosphate, a sugar called deoxyribose, and the nitrogen base. There are four bases: adenine, thymine, cytosine, and guanine. A, T, C, G. A and T bases pair up, and C and G bases pair up. The bases pair up forming hydrogen bonds. Let's talk about DNA replication. It's the process of producing two identical replicas of DNA. The first step is to separate the two strands by enzyme helicase, and results in the formation of a replication fork. The separated strands each provide a template for creating the new strands of DNA. The first strand is leading strand. Enzyme primase starts to make a small piece of RNA primer. Enzyme DNA polymerase binds to the primer and make the new strands of DNA. The other strands is the lagging strands. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strands. Let's talk about RNA. RNA is a nucleic acid found in the cytoplasm. RNA is made up from single nucleotide strands. Each nucleotide also contains three parts: a phosphate, a sugar called ribose, and a base. The bases in RNA are adenine, uracil, cytosine, and guanine. A, U, C, G. There are three different types of RNA: mRNA, tRNA, and rRNA. Firstly, mRNA stands for messenger RNA. It delivers a copy of the DNA code to a ribosome. Now for tRNA, stands for transfer RNA. It transfers amino acids from the cytoplasm to a ribosome for making proteins. The last but not least is rRNA or ribosomal RNA. It is the main component of ribosomes because rRNA bonds with special proteins to form ribosome. Talk about protein synthesis. A set of three bases in the DNA molecule codes for a particular amino acid. GGT, for example, codes for the amino acid called glycine. To get a code for the desired protein from the nucleus to ribosomes in the cytoplasm, a copy of the small ancestor section of DNA will be made. Protein synthesis has two steps: transcription and translation. I'm gonna talk about transcription first. The copying of the genetic code on DNA synthesis process and producing messenger RNA is called transcription, and it occurs in the nucleus. Transcription has three steps. First, initiation. In the initiation process, the RNA polymerase enzyme attaches to the DNA promoter, then assembles the RNA nucleotide and forms an RNA messenger chain. Second, elongation. In the elongation process, the RNA nucleotide arrangement process is along the entire DNA strand. RNA polymerase moves downstream to open DNA and extend RNA transcripts 5' to 3'. Third, termination. In the termination process, RNA polymerase transcribes DNA terminator. DNA, messenger RNA, and RNA polymerase separate. Let's move on to translation. Now we are going to translate that genetic code into something that the body understands by ribosome. and it produces polypeptide. It also has 3 steps. First, initiation process. In the initiation process, the small ribosomal subunit binds to the messenger RNA at the end of 5'. The carrier of AUG start codon as an initiation signal. Each of the 3 nitrogenous bases in the messenger RNA forms a codon that codes a specific amino acid. The codons UAG, and UGA are called subcodons or nonsense codons because they don't code for any type of amino acid. The amino acid binds to transfer RNA that has the complementary nitrogen base sequence with the codon that codes it. The sequence of nitrogenous bases is called the anticodon. Second, the elongation process is the addition of amino acids forming a complete polypeptide chain. Third, in the termination process, the ribosome reaches a stop codon reaching the A site or the amino acyl tRNA on the ribosome. The translation complex breaks down. Well, that's all from me. Thank you for watching. See you in the next time.